As we know, the seagrass meadows are very important along the coast. They provide valuable ecosystem services and functions. For example, they maintain high biodiversity, and as part of the biogeochemical cycling. In China, there are 10 genera and about 22 species along the east coast of China, from the temperate zones and to the tropical zones. But the seagrass areas has been declining globally. This situation also occurs in China, in the coastline. Due to Eurification, human disturbances, the continuous meadows has been fragmented into isolated patches. Many studies has demonstrated that the habitat fragmentation can decrease the population size, gene flow, and the reproductive efforts for the species. As a result, the population genetic diversity also decrease. And my question is, I want to know how the fragmentation affects the fine scale, spatial genetic structure, and the flowering output of Enhalus. To estimate the spatial genetic structure, we designed two plots with the size of 20 meters multiplied 20 meters in Hainan Island in the south of China. Plot A is in the fragmented meadows. The cover is 41%, and the plot B is in the continuous plots. The cover is 85%. The leaves were collected regularly according to the grade. With the interval of 1 meter, and the clone was identified by its microsatellite loci, and we also checked the male flower and the female flowers in these two plots from every month from June to December in 2017. Let's see the results. We found 214 genotypes in the fragment plots, and 255 genotypes in the continuous plots. From the maps, we can see that most of the genus of Enhalus was represented by one ramet, and the largest ramet genet we found in this study, and it has 15 ramets in the plot B, and in the red one. Overall, we found that there are more large clones in plot B than in plot A. This results suggest that the fragmentation may decrease the clone size of Enhalus due to the death of the shoots. Based on the analysis of spatial autocorrelation analysis, we found that significant spatial structure was found in both plots. within 2 meter and 4 meter respectively, both at the ramet-level and the genet-level. Here we can see the intensity of the SDS at the ramet-level is higher than the genet-level. This result suggests that the clonality of Enhalus increased the spatial genetic structure for Enhalus. We also compared the SDS pattern between these two plots at the ramet-level and the genet-level, and we found that at the ramet-level, significant differences between the two plots is significant, but at the genet-level, the SDS pattern was not significant difference between these two plots. Why the SDS pattern was changed at the ramet-level but not changed at the genet-level? We suppose two possible processes. First one, fragmentation may decrease the size of the genotypes, but here the genotype composition was not changed. Another process is that the fragmentation may decrease the genotype position, but after fragmentation, maybe there are still seedlings recruitment in this in the gaps. For the flowering output result, we found that the Enhalus exhibits luxury sexual reproduction. Although the flowers in plot A is slightly lower than plot B. Okay, here's the summary. In this study, we found that the fragmentation may decrease the clone size, reproductive efforts, and the ramet-level SDS, but not change the genet-level SDS for Enhalus. This pattern suggests that SDS at the ramet-level was more sensitive to the fragmentation, clonality, and the seedling recruitments might buffer the negative effect of fragmentation on the genet-level SDS for Enhalus. Okay, thank you.
That's some really nice research, well done, and it's so good to see you establishing the baseline pattern of clonal spatial distribution as a part of the first assessment. That's really, really good. I was just wondering about if you think that the original meadow established over a long time, or all with a mass recruitment all at one time?
A long time.
Yeah. So many recruitment events.
Oh, sorry. Could you repeat your question?
Before the meadow was fragmented,
Before, yeah.
Yeah, a long time ago.
Uh-huh.
Do you think it was a long-term persistent meadow that had built up over a very long time? Or do you think it was a meadow that fluctuated or changed a lot? Or don't you know?
The habitat here was disturbed in recent 20 years.
Okay.
Yeah. Before 20 years ago, here the Enhalus meadow is continuous and healthy.
Yeah.
Yeah.
So it's a well-established long-term meadow.
Yeah.
Yeah, great.
Okay.
Thank you, Shah. That was a really fantastic talk. Very exciting results. I had a question about flowering.
Okay.
So in the non-fragmented meadow, you said that the flowering was higher.
Yeah.
And then in the fragmented meadow, it appeared that the sex ratio was more skewed towards males, if I understood your graph correctly.
Yeah.
Was a little bit higher. Is that right? Were there significant differences? Did you test if there were differences between the number of flowers and also the sex ratio? Because it kind of makes sense that when the meadow is fragmented, they're investing more in male flowers so that the pollen has a better chance of moving and getting to a female flower?
Actually, this data is the total number for the plots for months, for 7 months. So that means one plot only has one data. So yeah, so no comparison. In fact, the total flower in the fragmented meadows is slightly lower than the continuous meadows, as you know, because the shoot, the depth of the shoot was more in the fragmented meadows. Yeah, but the interesting thing is that although the flowers decrease in the fragment meadows, but the output of the fruits is slightly high in the fragment meadows. So yeah, it's possibly associated with the pollination mechanism. Yeah. Okay.
Hi, sir. It's very well done. Yeah, I have a question for you. Do you have any replicates in two plots for each type of the meadow, fragmented and the continuous meadow? You have any replicates or just two?
Just the two plots.
Yeah.
Yeah.
Okay.
No replicate.
Yeah.
Yeah.
So, and another question is about the kinship value. You do found that the kinship value is very high in the very short distance, 2 meters.
You mean the interval?
Kinship value, very high in the very short distance. I saw somewhere.
Sorry.
The kinship value.
Oh, the kinship value.
Yeah,
okay.
Yeah.
Yeah.
Yeah, it's very high in the short distance. Yeah. So do you think it's relevant to the long potential dispersal of this species and Enhalus?
You mean why the kinship is high? Yeah. Okay. Usually, pollen or seed limitation was considered to be responsible for the SDS for the seagrass. But here, I think for Enhalus, the SDS maybe is caused by the cohort dispersal of the seeds or the pollen, not the limitation dispersal of pollen and the seeds. But I still need more data to support my hypothesis. Yeah.
All right, we might leave there. Please join me in thanking Xiao Yu and all the
Okay. Thank you all.